I'm really happy that I could attend this conference. It's been very, very illuminating. My lab uses a number of different microscopy approaches, but we sort of do everything in sort of very old-fashioned ways, human learning ways, I guess. But I also, as Steffi said, sort of like went back and forth on figuring out what to talk about, and I thought, well, I could spend like 40 minutes talking about my imaging woes and image analysis woes, but I decided not to do that. Instead, I'll tell you about this is a biological story. But everything that I've heard at this conference has certainly inspired me to try and move towards sort of extracting more information and from our experimental data. But for now, I'm going to switch into the mode of telling you about work in my lab on trying to understand how kinase signal is organized for cell division. So more broadly, my lab is interested in self-organization, as many of the groups in this room are as well. And the question is, how do we build these beautiful structures that are many microns in size from simpler building blocks that we now with both structural and biochemical assays, we understand quite a lot about these protein structures, their activities. But the question really is, how do we go from these building blocks to structures like this? And one of the things that I have focused on is trying to understand what are the collective activities of these proteins, what are the emergent behaviors, and can we build mini units, mini structural and functional units or modules of more complex structures like this to sort of connect the dots between these two length scales. So so again, like many people in this audience, the structure that is the inspiration for the work we do and for understanding principles of self-organization for us is the mitotic spindle. And this is a very simplified cartoon of a mitotic spindle. And if you remember from Steffi's talk, it is not what this looks like. There are many, many microtubules, but this is a simplified view just to kind of orient us to the steps of mitosis. Now, the end goal of mitosis is to form two equal daughter cells. What that means is that once the chromosomes have segregated, the cell has this task of finding its middle and pinching exactly through its middle to form two equal daughter cells. And this happens in anaphase when the site of cell cleavage is determined and proteins that are required for cytokinesis are selectively enriched in this region. So this then leads to the question of how is the site of cell division defined in dividing cells? So how do downstream proteins know exactly where this position is? So it turns out that the microtubule cytoskeleton plays an important role in this process. And in particular, there is a structure known as the spindle midzone that is assembled proximal to the site of cell cleavage at anaphase. And this structure is called the spindle midzone. And what the spindle midzone is an antiparallel array of microtubules that meet in the middle to form this. It's actually a very dense array of antiparallel microtubules, but the signature architecture of this bundle is that microtubules are in this antiparallel orientation, forming exactly at the site of cell cleavage. So I find the midzone a really interesting structure within the mitotic spindle because it seems to have both mechanical and signaling functions. So it has mechanical functions in pushing the microtubules apart, so in generating forces essentially to elongate the spindle. And at the same time, it also serves as a scaffold. So we can think of it as a platform that forms in the middle of the cell, which then can recruit these downstream regulators, such as kinases, regulators of GTPases, and so on, that are then required to regulate the machinery that then performs cytokinesis. Which then leads to the question that even if we have this bundle of microtubules, how are cytokinesis factors specifically recruited to this population of microtubules within the dividing cell? And there are two reasons, at least, why this is not easy. And I'll just tell you what they are using. Okay, so before I do that, let me actually quickly tell you a little bit about how this structure is built, the spindle midzone. And I'm going to tell you about a very minimal unit of the structure for the purpose of this talk. So in order to build this antiparallel array of microtubules, the protein that actually cross-links these microtubules is called PRC1, which Meredith also introduced in her talk. So this is a protein that cross-links microtubules with a high specificity for cross-linking it in antiparallel orientation. Now, this sets the geometry, this sets the antiparallel orientation, but this is not enough. As you can imagine, if the size of the structure is not correctly maintained and this structure is too wide, that would be a problem for positioning mechanisms. And therefore, the next thing that happens is that there are proteins that are recruited actually by PRC1 that are both sliding motors and proteins that regulate microtubule dynamics, and these pushing mechanisms, as well as mechanisms that regulate tubulin polymerization, control the length of the structure. Now, one of these proteins is this kinesin called KIF4A, and this is a protein that is specifically recruited to the midzone through interaction with PRC1. And together they form a minimal module, because this protein KIF4A can both slide microtubules as well as regulate microtubule dynamics, thereby controlling the width of the midzone. So we have one protein that cross-links and builds the antiparallel scaffold. It recruits another protein that controls length. There are many other proteins that contribute to it, but for today's purpose, we can think of these two activities as sort of the minimal components needed to build a midzone. But then this is all fine, but how do regulatory and signaling proteins recognize this structure? And let me just highlight why this is a challenging problem. So this is a movie I've taken from a paper from Thomas Surrey's lab. And when the timer starts, the cell is entering mitosis. In blue channel are the chromosomes, and in pink is PRC1. And what you'll see, and you'll see that right in this current state, it is decorating many of the spindle microtubules, but as anaphase progresses, you'll see it more heavily recruited on overlap regions. And I just want to play this movie to highlight how dynamic and complex sort of this process is. So there is the environment of the cell is constantly changing. The microtubules the bundles are getting more prominent there, and the cell is elongating, and further the bundles are also compacting. So within this very dynamic and changing internal environment of the cell, the downstream proteins that have to get to the spindle midzone, to this PRC1 marked zone, need to find these bundles and hop on these bundles. So the question is, how do proteins do that? So the other problem is that the spindle is made of a large number of different subsets of microtubules. So there are kinetochore microtubules, there are astral microtubules, there are these large interpolar microtubules, and then there are midzone microtubules and more. So all this to say is that there are many different subpopulations of microtubules. And again, this is a challenge for proteins that are trying to get to a very particular subset of microtubules at the cell center. So therefore, we sort of like, and this is part of the reason why I find this question really interesting. How do proteins find the right subset of microtubules? What are the molecular mechanisms by which these regions are specifically where proteins end up? So I'm going to tell you about our work in trying to beginning to understand this question with a focus on an essential mitotic kinase, Aurora B, which Vijay had in her talk. And the reason is that it is an important mitotic kinase. It's important for cytokinesis, and it's one of these downstream signaling proteins that is enriched on the spindle midzone in anaphase. So at metaphase, the Aurora B kinase is on the chromosomes, and then upon anaphase onset, it is observed to specifically relocalize to this bundle of microtubules, which I've just represented here by a pair of antiparallel microtubules, and concentrate in this region where it then phosphorylates downstream proteins that are required for cytokinesis. Okay, and one of the key proteins that is required for removing Aurora B from the chromosomes is a kinesin, and it's called MKLP2, and I'll come to it later. But before I start talking about mechanisms, let me just introduce you a little bit more to Aurora B, because it actually comes as part of a complex. So the CPC that's the Chromosome Passenger Complex is what contains this mitotic kinase Aurora B. And the protein scaffold for this complex, so to say, is this protein called NSF. It is this really large protein. And at the C terminus of this is the Aurora B binding and activation sites. So Aurora B binds to this site and it is activated through this binding. So at the C terminus is the kinase activity. At the N terminus, there are other proteins which are then involved in chromosome binding, but also microtubule binding. There is also a second microtubule binding site present within the tether. So this is a complex that has a lot of function. It can bind DNA, it can bind microtubules, it binds Aurora B. So it's just got all of these different activities. And this tether is actually quite interesting and very poorly characterized. Because of its size, this protein has just been really challenging to work with. But this long tether is composed of an intrinsically disordered domain. So this is the IDR. And there is a single alpha helix that forms part of the structure that's proposed to have spacer functions within this complex. So example, you can imagine that if this kinase complex is sitting on the chromosomes, then there are models that propose that this tether is sort of acts as a distance setter to determine how far the Aurora B kinase can go, and so on and so forth. But essentially, this is a long tether that sort of sits between the two ends of the molecules. So we wanted to address this question of how the CPC is enriched at antiparallel overlap. So we decided to sort of really simplify this question and ask, can we build this in vitro? Can we actually reconstruct this geometry of two antiparallel microtubules where CPC enriches on this overlap region of the two microtubules? And the approach that we decided to use is biochemical reconstitution. But we immediately had a big problem because we to do a reconstitution, you need proteins. And the recombinant human CPC was a big challenge to purify. And then the second thing we need is an assay, which we had developed in the lab for other studies. So this is the work of Costas in my lab, who's a postdoc who really took on this task and just didn't give up on this purification. And I won't go into any of the details except to say that he got this beautiful purification of the human CPC. It's the full-length complex, and it has all of its parts, the microtubule binding regions, the kinase, all of the parts are intact. And we were finally able to start doing reconstitution studies connected to its microtubule binding activity. I think this is also the first SDS paid gel of this conference. So, so I'm just going to take a little bit of a detour and tell you some structural characterization we tried to do of the human CPC. So, one of the things we tried immediately once we had this beautiful complex was ask one of our cryo-EM friends to put it on a grid and see what it looks like. But as you'll see, because of the structure, because of the properties of this complex, it was actually, it's still a work in progress. This is going to be a harder problem to solve. But meanwhile, an amazing former postdoc in my lab, Sitara Vijayaratne, had actually developed some atomic force microscopy imaging in the lab to actually look at microtubules. And she said, I want to look at this complex and see what it looks like. And so that's what she did. So what we did then is we modified the CPC to sort of put two signal amplifiers at the two ends of the complex. So we put a GFP tag on one end and a halo tag on the other end. And other people in the AFM world studying intrinsically disordered proteins have done this. And what this does is just amplify the height of the molecules at the two ends and gives a nice signature that makes it really good for imaging. I won't go into AFM imaging as a technique, but essentially it's a cantilever with a small tip that is oscillated along a surface. When the tip hits an object, it is deflected, giving a readout of the height. It's extremely sensitive to height, and what we get at the end of the experiment is the surface topography. So what do these molecules look like when we look at them on a mica surface by AFM? What we noticed immediately, and we were very happy that that was the case, was we saw pairs. We always saw these molecules come in pairs. And the other thing we noticed was they were separated by different distances. Now, because the signature of the GFP and the halo are different, in these sort of still images, when we do very slow scanning, we can actually pick out where the N and C terminus are in many cases. And then depending on how the molecule is oriented, we can also see these tethers. So what these data immediately told us was that we have these intact tetrameric molecules. We can observe them, and this tether can actually take on potentially different lengths, or at least we saw different lengths. And we thought maybe this is a very conformationally flexible molecule and that we could measure its intramolecular dynamics. And so in order to do that, what Sitara did was she did some high-speed AFM imaging. And I'll show you some different types of molecules in this grid. So now we are doing fast imaging. It's at 100 hertz. So here's a molecule that's stuck. And you can see this tether sort of fluctuating between the two stuck ends. Whereas here's a molecule that's not stuck. And you can see one end moving relative to the other in time. And it actually gives me, from these kinds of data, in dark brown is sort of these distances that the molecule explores. And we've combined the distances from all of the molecules. And what we saw in this case was this sort of large peak at these longer distances that are about 80 nanometers or so, showing that that's sort of the distance of the molecule, but it can span a whole range of distances less than that. If we remove the tether and do these experiments again, we no longer see these large distances, and all of the intramolecular distances are now short. So the way we analyze this data is entirely manually, and it's actually very painful. And I'm sure we are losing a lot of information. And so this is one of the kinds of experimental data we have where I would love to have a more automated pipeline. So, and that was the main reason I wanted to show that part. So we have now recombinant CPC, and now we can get back to our biological question of how is CPC enriched at antiparallel microtubule overlaps. So how do we actually reconstruct these types of mini modules in the lab? And the assay we use is the following. So we adhere single biotonated microtubules to glass cover slips using Neutravidin linkages. And this is, in this case, it's labeled with a 488, and you can see the image of a single microtubule on the right. We can then add any microtubule bundling protein of choice. We add it, we let it decorate the microtubules, and then we add more microtubules which are not biotonated. So they cannot bind the surface, but will only bind when it can form a bundle. So here is an example of what happens when we form these sort of microtubule sandwiches. And what we have here is a pair of two microtubules. It's cross-linked with this particular protein, and from here we can get overlap regions, and we also get a readout of how the protein binds on the single microtubules, and we can quantify essentially enrichment of protein on overlap regions relative to single microtubules, and that's the assay we're going to use. So what are the proposed mechanisms? CPC, it's been observed for decades now that CPC is at the spindle midzone, and we're not the first people to ask how does it get to the midzone. So what are the models that have been proposed? So one model is that there is the intrinsic microtubule binding and bundling of its CPC. Remember, I told you CPC has two microtubule binding domains. Its intrinsic microtubule binding and bundling contributes to its microtubule, to its enrichment at overlaps. But I won't go into some of the details why we think this may not be the whole story, so we wanted to test this. The second is this kinesin, which removes it from CPC. It's a motile kinesin, and subunits of CPC can be carried by MKLP2 along microtubules. So there was this like really, I think, solid mechanism where it is a transport-based mechanism that carries the CPC to the plus end of microtubules. So transport is fine, but how does that then lead to enrichment at these types of structures, at antiparallel overlaps, was unclear. So we wanted to look at that as well. And then finally, this protein PRC1 recruits multiple different proteins through direct interactions. And so we also wanted to test if, in fact, CPC got to the midzone through interactions with PRC1. So I'm going to show you one piece of data here for what experiments like this give us. And essentially, what we have here is in this assay, where we are now asking, is the CPC bundling sufficient for midzone localization or overlap or localization or antiparallel overlaps? So here is our sandwich. Here is one microtubule, and here is the other microtubule, and between these white lines is the overlap region. And then when we looked at the CPC signal on these bundles, you can see that in this bottom image here, and there is absolutely no specificity for overlap. So CPC does bundle microtubules very well. as has been previously shown, but there is no selectivity for overlap over single microtubules. It just binds everywhere. We did this experiment over a whole range of CPC concentrations, and we still did not see any specificity. So we basically rule out this mechanism, or we don't think it's probable or sufficient for CPC's midzone localization. And using the same assay, I'm not going to show you all the data in the interest of time, but using the same assay, we were able to show that again, the MKLP2 trafficking models doesn't quite work, and neither does PRC1 interactions of either CPC or MKLP2. So none of these proteins, which we painstakingly purified in the lab, gave us the answer. So we kind of—
These are all stable microtubules, right?
These are all stabilized microtubules. Yeah, and we just did that for keeping the assay simpler. Yeah. So what is the missing factor? So we kind of were back at the drawing board, and essentially it was suggested that a different kinesin may potentially be involved in Xenopus oocytes as a CPC transporter on astral microtubules. So we thought, okay, let's look at that kinesin. And that kinesin is a different mitotic kinesin. It's KIF4A. This is the protein that is recruited by PRC1 to the midzone. This is the protein that I introduced as the protein that regulates microtubule length. So we did this experiment now with PRC1, and so we build the bundle with PRC1. We add KIF4A and we add CPC. KIF4A is dark in this experiment. We are only imaging PRC1, CPC, and the microtubules. But as you can see already from this image, so here is now a bundle of microtubules. Here is PRC1 beautifully enriched on this antiparallel overlap. And then here is CPC. Doesn't look bad at all. So we were very excited because when we calculated ratios of overlapping to single microtubules, with all of the other components, we are here in terms of our median, and we got almost 5-fold enrichment the minute we added KIF4A to our reconstitution. But we noticed that, while there is nice enrichment at this overlap, there are also molecules that are just sitting on the outside, still not quite getting to this overlap. So we said, what else can,
So, but you do require the PRC1 if you just made overlap in another way. So it's not independent of PRC1, right?
So, so we didn't look at that too carefully because this is the most relevant system. But even with KIF4A alone, we do get CPC enrichment. Yeah. So it's not technically, I think, entirely PRC1 dependent, but I think this is the relevant system, to look at.
And the stoichiometry is 1-to-1 when it comes to PRC versus the other component like PRC.
So PRC1 is at much lower levels here relative to all of the other proteins because it is a highly cooperative crosslinker, and it enriches in these overlap regions at very low concentrations, and it can bundle at very low concentrations. Yeah. So, okay, so we said, well, we just kind of did this experiment for fun. So we said, well, we now have these other kinesins, so let's add that in. So we basically did this experiment where we now have CPC, PRC1, KIF4A, and MKLP2. And then we got just the most beautiful enrichment on overlaps that we had seen of all the conditions. And here is now the quantification. And here now we can almost see a 10-fold enrichment of CPC on overlap regions relative to single microtubules. And this is the level of enrichment that we see with PRC1 alone. So this is really getting to very high levels of specific enrichment. So we were very excited because we finally had a system. We were finally able to build a minimal system, a minimal module where we could get this mitotic kinase complex to regions of overlap that were formed by midzone proteins. Okay, so what I've shown you so far is images from steady state. So I just want to show you a couple of movies for how the system emerges and forms. And it's a little bit complicated, so I'll walk you through it first using this schematic. So we have these two microtubules. In green, dumbbells are the PRC1. We add the CPC and the kinesin. So when we add the kinesin, the microtubules move, and then they actually eventually stall. And we are asking, where does CPC go in this sort of sliding system that eventually stalls? So this is the kind of movie that you'll be seeing next. And there are only, again, what is labeled is PRC1, CPC, and microtubules. And I'll walk you through this too. So this is now movies from these experiments. This is with KIF4A. PRC1 is in green. In the bottom panel is the CPC, that's in grayscale, and magenta is the microtubules. And as I play this movie, you'll see the overlap shrink, and the green PRC1 really concentrates at the overlap. And the question we are asking is, how does CPC follow and reach that same position? So as you can see here, the CPC zone of antiparallel bundle is getting smaller because of sliding. And as that happens, the CPC is concentrating. But you can see that there are molecules that don't quite make it. They just end up on these outside regions, hanging on and not quite getting to the overlap regions. So
also see PRC at the same place?
Yes, so the green is PRC1. So you see PRC1 over there. There are some, so when we see small pieces like this, that's really caught a very good thing, though it's probably a very small piece of microtubule. So it's not, there's just another like piece of tubulin cluster or bound to it. So it's not, yeah.
The only processive part is KIF4, right? Like
only active protein, yes, is KIF4A, yeah. But I'll play the movie now with KIF4A and PRC1. It's a little bit more complicated. But the main point here is, unlike this movie on the right, that one you can actually see really robust movement of particles that are outside the PRC1 zone being moved to the overlap region. So here there is the presence of the second motor essentially takes the molecules that are sitting outside and really brings them all to the midzone type overlap region to concentrate them. And this two-motor system works because of that. One motor really concentrates where PRC1 is, brings CPC to it. All the proteins are concentrated there, and the other motor does a better job of bringing proteins from the outside and concentrating them to this overlap. So in the spirit of this meeting and part of my image analysis woes, I just want to show you what the field of view of an experiment like this looks like. So we kind of go through every single bundle one by one to find ones where there are pairs. And then we make sure that these are not like non-specifically stuck. And so these are the yellow ones are the ones that we can pick out by eye and we can do it. And it really takes a lot of training to spot them. But then there are a lot of structures like this that I've highlighted in green that are multi-microtubule cluster. They're bundles of bundles. And in fact, we are not analyzing them. And I think we are throwing away really probably interesting information on how larger structures are organized. And then there are single microtubules where these proteins all accumulate at the end. So there are different populations and just to keep sort of some focus and be able to do the analysis in a reasonable amount of time, we are simply looking at just the two microtubule structures here. So this is the kind of, this pains me. I feel that we should be doing better.
Do you control if they're antiparallel or parallel?
That's a great question. So in this particular experiment, so when we have PRC1, we know from other experiments that our bundles are typically antiparallel. So when we make our sandwich with PRC1, we know the bundles are antiparallel. Also, the sliding microtubules are antiparallel. But the experiments where there was no sliding or there was no PRC1, we don't know the polarity. We analyzed all of the bundles, and we just we don't see two populations, but we are in the process of also doing experiments with polarity marked microtubules to sort of verify and validate some of those results. Yeah. Okay. So so what our results showed us was that this kinesin KIF4A is actually playing a pretty important role in our in vitro reconstitution assay. It's absolutely necessary in our reconstitution assays to get enrichment of CPC at antiparallel overlaps in vitro. But what about in cells? Is CPC important for CPC is KIF4A important for CPC localization in anaphase cells? Now, this experiment is technically challenging for biological reasons, which is that when KIF4A is depleted, the spindle midzone, as marked by PRC1 in this image, is wider. So you just have these wide midzones. And so all of the proteins on these microtubule bundles are just dimmer. So it's really hard to know whether any defects in localization are because of structural defects of the spindle, or is it because of recruitment defects? So we thought that perhaps we could try and address this by doing correlation analysis. And this is the logic of the experiment, which is that if we look at CPC on these wide midzones, if we see a clear correlation between PRC1 signal and CPC signal, that would indicate that the recruitment is independent. Even if the signal is dimmer, the localization would be correlated. Whereas if it is KIF4A dependent, then that signal would be lower. So that's the experiment we did. And just in the interest of time, I won't go through all of the images, but this is what wild type control cells look like. And there's a very nice correlation between the PRC1 channel and the Aurora B channel in each of these bundles, which we also analyzed manually, because picking out these bundles in the midzone actually turned out to be very tricky by any other method. However, in KIF4A depleted cells, we see a clear loss of Aurora B signal from the midzone microtubules. And in fact, we see an increase in the cytoplasmic signal, suggesting that there is a loss in this correlation, and therefore KIF4A is a molecular anchor for CPC at the spindle midzone. Okay, so in the last few minutes, I just want to address one question, which is, if we think of CPC as a cargo, and it's being moved by motors, why use two motors? So just to kind of zoom out and talk about other cases in cell biology where transport is mediated by two motors, there are examples like bidirectional transport, where same cargo is moved to the plus and the minus end, and it requires different motors. There are cases where it's been proposed that for example, on a vesicle, by putting many kinesins, you can go around roadblocks. And here we have a system where potentially it looks like using two kinesins might be useful for sort of navigating these different geometries, like regions where the microtubules are not overlapped and where they are overlapped. But we were like, is that really what's going on? So I'm going to skip this. So we decided to do this experiment in an even simpler way. So we decided, okay, let's get rid of this bundle altogether. And let's not have PRC1 and just do this experiment with the motor and CPC alone. So here we just now have single microtubules on a glass cover slip. There's a motor, it's carrying CPC, and we simply look at its transport properties. We make chymographs where on the y-axis will be time and x-axis will be distance, and all of these diagonal lines represent protein trafficking. And in these experiments, we are only labeling CPC. So we are now looking at chymographs of fluorescent CPC on a microtubule at different concentrations. So here when we have, so now we first did these experiments at nanomolar concentrations of CPC. And we ask, so in the presence of KIF4A, you can see all of these diagonal lines and the proteins accumulate at the end. So it's very robust trafficking. However, if we now look at this, the MKLP2 channel experiment, now we can see that basically you see all of these straight lines, which suggest that there is no effective transport. So the molecules are not getting moved by MKLP2 under these conditions. However, we notice that very early on, if we look at the first 2 minutes or so, we see some transport. And we were like, okay, it's not that the MKLP2 is not able to transport CPC. We know it can. We know from our other experiments too. But it is somehow dependent on the density of the CPC itself. So maybe in this initial time point when the molecules are just binding, there's less crowdedness on the microtubule. And so the trafficking is fine. And then at later time points, it stops. This was a hypothesis. So what we did was an experiment where we changed this concentration of CPC from the nanomolar regime to the picomolar regime. So it's a true single molecule experimental regime. And what we see now was remarkable. We now saw these beautiful transport of CPC by MKLP2. We can see all of these molecules now being moved. And in the presence of KIF4A, we still get movement, but we see more diffusive movement as well. So what this says is that the two motors, so now we don't have geometry, we don't have PRC1. We are simply looking at CPC transport, and the two motors are just better tuned in different density regimes. So when the CPC density is higher on the microtubule, the KIF4A is a better motor. When it's low, then MKLP2 is a better motor. And I think this is how the system essentially works to concentrate the protein at the midzone. So I'm not going to go through this. We also tested this hypothesis by changing CPC's microtubule binding affinity, and it led to the same conclusions. So I just want to wrap up by summarizing the reconstitution and what we find to be a minimal module for concentrating a mitotic kinase complex specifically to overlap regions. So the first component here is PRC1. It cross-links antiparallel microtubules. It has a very high affinity for antiparallel microtubules and strongly enriches in that region. It then recruits KIF4A through direct and strong binding interactions to this region of overlap, where it has an important role in regulating microtubule architecture, but we find that it is a direct CPC binding protein, and it actually concentrates CPC as well at that overlap. On the other hand, there is this other important module composed of CPC and MKLP2. This is required for getting the CPC off the chromosome. This is a high affinity interaction again. And these, in the context of recruitment to the midzone, we think this plays a role in bringing molecules that are sort of left on these non-midzone microtubules to the center. And what is really interesting is that these two modules, the PRC1 KIF4A module and the CPC MKLP2 module, are connected by this lower affinity. I didn't show you the data, but this affinity is lower through a lower affinity interaction, which is essentially offset by the high concentration of these proteins that enrich at the center. So what is really interesting here is that all 4 proteins can bundle microtubules. And there are these but the system works through a hierarchical sort of series of interactions that are characterized by very different binding affinities. And I think from sort of like understanding and like trying to get at the mechanism, a principle that sort of falls out that was, I think, really interesting to me is that this is a system where there is adaptation to local cargo density. So where the cargo density is high, you use one motor, and where the cargo density is low, another motor is better tuned for transport. And in the context of a structure like a spindle, especially like a structure like the spindle midzone that is evolving over a time scale of minutes during anaphase, I think these types of adaptive mechanisms probably allow these downstream proteins to precisely localize and bind the specific subset of microtubules and concentrate there. So for example, in the context of the midzone, these regions are regions of high cargo density, and these would be regions of lower cargo density. And we think this is why the two-motor system is potentially the best solution for, maybe not the best, but a better solution for a problem like this. So with that, I'd like to thank the people in my lab. I'd like to particularly thank Costas. I think his perseverance really was the reason why we were even able to start working on CPC. He's an amazing biochemist. And then I want to thank Sitara, who really sort of set up all of the AFM imaging work in my lab. And the rest of the lab, they're a really great group. So thank you all.
Time for a question or two?
So if I remember correctly, you showed that these two proteins, MKLP2,
Yeah, it doesn't matter.
Two proteins, yeah, they have differential binding affinity to CPC based on CPC concentration, right?
The binding affinity is the binding affinity. That doesn't depend on concentration.
Yeah, there should be two independent parameters.
Yeah, so I didn't show that data, but KIF4, actually MKLP2 has is tighter binding. I don't remember the numbers, they're on my slide. I think one is they're about 5 or 8 times different. So KIF4A has actually a weaker binding affinity. And I think that is advantageous because CPC is recruited directly by KIF4A to regions where KIF4A intensity or density is the highest, and that's the midzone. So I think that lower affinity actually is better for specifically bringing CPC to PRC1 marked antiparallel bundles.
How do they sense the concentration there? Like CPC is binding there, whether it should bind to MKLP or KIF4, how is that regulated?
So I think CPC can bind. So CPC can bind KIF4A and it can bind. So once CPC is removed from the chromosome arms, it's going to see a population of KIF4A where PRC1 is, and that's where the that's a fraction of the CPC molecules, let's say, have bound there. CPC is also a microtubule binding protein, so it will tend to bind in other places that are outside that zone. And MKLP2 potentially transports it into the region. Yeah. Yeah.
So, how does binding more CPC kind of, tell the activity of MKLP?
Yeah, that's a really good question, and I wish I had an answer. So I can tell you what might be happening. I think one thing is that MKLP2, so different motors deal with roadblocks differently. Some can walk around, some dissociate, some get stuck. So I think MKLP2 must be one of those molecules that cannot walk effectively if its road is too populated or if the occupancy of the microtubule is high. Yeah. Yes.
And toward the beginning of the talk, you showed one primer whereas the PRC1 is at the midzone, but then the PRC1 signal is sort of constricting like this. It
Yeah.
reminded me of like actin contractility, like is something gets contracted in there, or
I mean, that's the actin-actomyosin ring.
okay, so the PRC1 is on the actomyosin ring?
No, it's a separate, the whole cell is sort of getting,
I see.
the center of the cell is getting contracted.
One more quick question.
I have two very quick questions.
That's fine.
One is that from like Stephanie's talk a couple of days ago, potentially the microtube, they're not spanning the whole cell, a lot of them. So would you imagine that CPC is sort of everywhere, but only the ones in the region that is like one microtubule away from the midzone are able to collect there? If they're being transported by like you can't then collect anyone who's not on a microtubule who's close to the midzone.
So the ones that are, so I think the ones that are away are brought by the different motor. So the microtubules in the middle that are antiparallel, I think the one of the motors is sufficient to.
But you think they could hop over different microtubules?
I think it's possible because they're all dimeric, or they all have multiple microtubule binding domains. So I think it's possible that they hop over.
The other thing
But it's also just, even if they don't like necessarily hop over, if they dissociate close to the region of the midzone,
Yeah.
there is just a high microtubule concentration.
And do you think the speed of the motors versus like the cell size is something that could set in this mechanism, set kind of the timescales of cell division?
Yeah, we haven't looked at any of that at all, and I think we should do that. Yeah, we haven't. This is where I think we should do some modeling to figure out like how the motor velocities and binding affinities. We have all the data, but we just haven't done that.
Yeah. All right. Let's thank Radhika again.